Good morning, everybody. So, I don't know what's happening over in Keene today, but I'm over in Milford. It's about 9:45 in the morning, and the snow is coming down at a pretty decent clip today. So I know that classes aren't canceled over there, but if you are traveling around or traveling in the east, know that it is snowing pretty heavily already in Milford. So please be careful out there. I want to start off today's lecture by just saying that yesterday was an important day in history. Yesterday was Darwin's birthday, and so a couple of us in the Science Center put up a little cardboard cutout of Darwin. He'd be 205 years old today, and so we had a little mini celebration in the biology department yesterday. What I want to do is I'm going to start off by really just reviewing Darwin's four postulates again, and then we're going to dive into the structure of DNA. And know that this is a very superficial description of synthesis, transcription, and translation. And that's mainly because you get a very detailed description of those processes in other classes within the biology curriculum. And so we're not going to focus a lot on those particular detailed aspects of synthesis, transcription, and translation. But also because what's really important for this course and for evolution is understanding the results of mistakes that can happen during DNA synthesis and the cell cycle, and how it is that those mistakes can influence the characteristics of organisms, particularly in the proteins they produce and then ultimately in their traits. And so let's start off by just reviewing Darwin's four postulates. So if you want to review this on your own, you may want to pause the video really quickly and just sort of in your head state those postulates. It's really good practice for you guys. So the four postulates are the individual organisms that make up a population vary in the traits they possess. The second one is some of the trait differences are heritable, meaning that they're passed on to offspring genetically. And then the third postulate is in each generation, many more offspring are produced than can possibly survive. Thus, only some individuals in the population survive long enough to produce offspring. And among those individuals that produce offspring, some will produce more than others. And then the fourth postulate is the subset of individuals that survive best and produce the most offspring is not a random sample of the population. Instead, individuals with certain heritable traits are more likely to survive and reproduce. Now, we're all very familiar with these postulates at this point, but in order to really understand evolution by natural selection, we have to understand these two postulates very, very, very well. Okay, particularly the second postulate, which talks about heritability. And in order to begin to study to begin to understand heredity, we have to understand the structure of DNA. So I'm going to give you a little brief lecture about the structure of DNA, how it's copied, and how it is that those copies are transcribed and translated to produce proteins which impact the traits that we all possess and that all organisms possess, from the smallest bacteria to the largest mammal. I also want to emphasize that, again, this lecture is pretty superficial, focusing on the aspects of synthesis, transcription, and translation that are really important for evolution. But there are additional excellent resources that go into the details of synthesis, transcription, and translation. One of those is your textbook, and then the other one is this website, which is learngenetics.utah.edu. And I highly recommend that if you want to, if it's been a while since you've had some information about DNA structure, transcription, and translation, read your textbook carefully, but also check out this website. I really like a lot of the resources that are available on that site. So let's talk about the structure of DNA and really more broadly about the structure of nucleic acids. Okay, so DNA is a nucleic acid. It is deoxyribonucleic acid. But there's also another nucleic acid, which we're all familiar with, which is ribonucleic acid, or RNA. Nucleic acids are made up of nucleotides, and each nucleotide that is a component of a nucleic acid is made up of a phosphate group, which really doesn't change in structure, a 5-carbon sugar that differs depending upon whether you're talking about RNA or DNA, and that sugar differs really in the presence of a hydroxyl group at the 2nd carbon of the sugar in RNA, and the presence of just a hydrogen at that 2nd carbon in DNA. Okay, so functionally, the primary difference in the sugar molecule between RNA and DNA is the presence or absence of that hydroxyl group. The 3rd component of a nucleotide is a nitrogenous base. And in DNA, those nitrogenous bases are cytosine, thymine, guanine, and adenine. These are the nitrogenous bases. In RNA, those nitrogenous bases are cytosine, uracil instead of thymine, guanine, and adenine. Okay, so these are the basic structure of any nucleotide, and there are many nucleotides that make up a nucleic acid. So nucleic acid forms when each of these nucleotides consisting of a sugar, a phosphate group, and nitrogenous base are linked together through what are called phosphodiester linkages to one another. So if you look here, you can see that there is some sort of a pyrimidine attached to this sugar, which has a hydroxyl group, which means that it's ribose. Okay, and then the phosphate group. Here's the phosphodiester linkage, linking another sugar with its phosphate group and its nitrogenous base, which in this case is a purine. Now I'm going to go back for a second. If you look, nitrogenous bases are placed into 2 categories. They're either pyrimidines or they're purines. And these 2 different types of nitrogenous bases really just differ in their chemical structure. So A and G are purines, cytosine, uracil, and thymine are all pyrimidines. Okay. The way that I always remember what's a purine and what's a pyrimidine is I always think agriculture, or AG, is the purest form of living. And so that's how I always remember the differences between which nitrogenous bases are purines and which are pyrimidines. So here you can see a strand of RNA consisting of a pyrimidine and a purine. Okay, and what happens is that these phosphodiester linkages link together several different nucleotides in a long strand. So if you look at this single-stranded RNA, which RNA is single-stranded, you see the ribose sugar with the hydroxyl group, and you can see here's one nucleotide, here's another nucleotide, so forth and so on. Now, the way that RNA and DNA is read is essentially by reading what nitrogenous base is a component of each nucleotide in the nucleic acid, or in the nucleotides that are linked together to form the nucleic acid. So you can very easily read this RNA molecule. Okay, so here are the structures of the purines and pyrimidines that are nitrogenous bases for nucleic acids. This is RNA. So we see that the sequence is U, right? So U, A, G, and C. So you can see the how it is that nucleic acids are read. Okay. Now, DNA is different from RNA. It possesses the nitrogenous base thymine instead of uracil, and DNA is double-stranded. DNA and its primary structure, and the way it's depicted in many textbooks, is pretty much consists of a picture of this sugar phosphate backbone, which are sort of these red arrows here, and with the nitrogenous bases linking to one another. Okay. Now in DNA, what happens is nitrogenous bases link together purines to pyrimidines. Okay. So A always, which is a purine, always forms a bond with T or thymine, which is a pyrimidine. Okay. GC, GC. Okay. The way it is that these chemical bonds occur between these nitrogenous bases, again, this is sort of your sugar phosphate backbone. So the way it is that these bonds occur between these nitrogenous bases actually cause the DNA to form a spiral or a helix, which is really the secondary structure of DNA. So it is how it is that these hydrogen bonds form that determines or that causes that spiral in DNA. Again, here is your sugar phosphate backbone. Here is your sugar phosphate backbone. And here you see purine forming hydrogen bonds with pyrimidine. Okay. Here you see a purine forming hydrogen bonds with a pyrimidine. Okay. So that is the basic structure of DNA. Now DNA, which is what we're going to focus on here for a little while, is, again, a double-stranded molecule which consists of letters. And the DNA strand, if you just take one strand, is made up of letters. And if you think about it, these letters each make up words that consist of a triplet of nitrogenous bases, okay, or a couplet of nitrogenous bases. And so each of these words then make up sentences, okay? And so here you have a sentence of DNA sequences. And these sentences you can think of or should think of as genes. And each of these genes will code for a particular protein, okay? So DNA strand is made up of a series of letters, which are the nitrogenous bases. The nitrogenous bases make up words of 3 letters each, okay? And each of these 3 letters are different sets of these series of words that are made up of 3 letters make up sentences, okay? And then these sentences code for proteins in our bodies. And those proteins are what determine our traits. Okay. Now, what's important to think about in thinking about what happens in cells and the passing down of traits from one organism to the next through reproduction is you have to think about the cell cycle. Now, each one of our cells, which contains DNA and also RNA, undergoes a cell cycle. Okay, so we have cells that start off in what's called the first gap phase. In this first gap phase, you have largely the cell undergoing general function. Okay, so proteins are being produced, and that cell is sort of responding to the demands of the organism by producing proteins that allow an organism to react or have a particular trait. If that cell is then going to undergo division, what it does is it moves into what's called the DNA synthesis phase. Excuse me. And it's during DNA synthesis that DNA is copied. Once that DNA is copied, the cell has to prepare to undergo some sort of division. In this case, we're talking, the division is mitosis, but this could be meiosis. But since we're going to be talking about meiosis next, we'll just talk about, mitosis next. We're focused on mitosis here. So DNA is synthesized or copied. That cell then undergoes preparation for division. The cell enlarges, organelles are produced, particular proteins, so some transcription and translation is going on here. to produce particular proteins that are important for division. And then those copies of DNA are then distributed into 2 daughter cells, both of which then undergo or have the potential to undergo the cycle again. Okay, a lot of cells stay in this first gap phase for a very long period of time. So some of our heart cells, for example. Some cells in our body undergo the entire cell cycle very, very quickly and pretty constantly. Think about maybe a skin cell. Skin cells are constantly being lost and need to be reproduced. And so they are constantly undergoing this cycle. The G1 phase is generally twice as long as the G2 or second gap phase, but the synthesis phase is also one of the longer phases of the cell cycle, with the cell division phase, in this case mitosis, being very, very short relative to the other phases. Okay. What we're really going to be focused on in this section of the lecture is this phase, the synthesis phase. So here we have a cell that is in the first gap phase, and it is about to undergo a division, and so the DNA has to be copied so it can be distributed into 2 daughter cells. Okay, so let's talk about what happens during synthesis. Now, during synthesis, what has to happen is the DNA has to be, the 2 strands of DNA have to be separated from one another. And then new nitrogenous bases have to be linked to each of these separated strands of DNA, okay, to create 2 identical copies from one. Now, if you were in Cells and Molecules, or if you remember your high school biology, you know that synthesis is actually quite complex, involving quite a few different enzymes, proteins involved, that there are differences between the way the leading and lagging strands are synthesized. You have Okazaki fragments involved. We're not going to go into all of those details. We're going to really look at synthesis at its most basic level. During synthesis, what happens is a DNA molecule is with 2 strands is separated in a DNA molecule which consists of 2 strands of DNA that are linked together via hydrogen bonds are separated from one another. Okay, so that you have 2 separate strands of DNA. Here's one, here's one strand and here's the other strand. Okay, and then each of those separated strands is copied, producing 2 identical copies from one. Okay, those are the basics of DNA synthesis. That's the level that I want you guys to understand for this class. You can play around with this synthesis piece a little bit, and it's, again, in its most basic form. There's this really great website that is called Synthesize This, or is a part of this University of Utah website. Sorry, I'm just going to have to resize this so you guys can see the entire screen. And so you can actually practice building your own DNA molecule. So it'll take you through what happens during synthesis. So here is a single spiral strand of DNA, and you can literally build, by linking the appropriate purine to the appropriate pyrimidine, a DNA molecule. Okay, so if you want to practice synthesis, you can go onto the site and practice synthesizing your own piece of DNA. Okay, I'm pretty slow at it. This actually happens very, very quickly in the cell, and in most cases it's amazing because it happens so quickly in the cell that that more mistakes aren't made. So if you want to practice sort of synthesizing DNA on your own, it's a really nice visualization of how it is that DNA is synthesized. Okay, Now, but what's important is that it is during synthesis that mistakes are made. Okay, so you may have DNA being synthesized or copied, and then what happens is instead of the polymerase or the enzyme grabbing a T, which normally would pair with an A, it might grab a C, okay, and then continue to build correctly. A, G, C, G, A, okay. In most cases, these mistakes are corrected by a special enzyme that repairs mistakes pretty quickly. But it's when these mistakes persist after synthesis that is when things become interesting. Okay, so it's during this process that we often have mistakes or point mutations happening. And so many of these mistakes are repaired by enzymes. But what if they're not? And what do these mistakes actually do? Okay, this is where I'm going to end it, and I'll start the next YouTube video from here.